So allopatric speciation is defined as speciation, or the formation of new species from previously existing species, which is caused by separation geographically, or at least initially caused by that. And it's the way that we believe that the finches that Darwin described on the Galapagos Island probably evolved. Genetic data has confirmed that the species of finches on the Galapagos Islands are very closely related, and that they came from a single ancestral species of finch that migrated from South America to at least one of the Galapagos Islands at some point in history. And then as that single species populated one or more islands, additional birds, or birds from that group, moved over to other islands, especially as they were forming. They're all volcanic islands, so they're newly forming during this time, and that the various the birds moved to different islands and then started to experience different environments, maybe different mutations, maybe some genetic drift because of the small group that moved to that island. And so a combination of factors led to the different groups speciating into what are now closely related species. And so allopatric speciation is any kind of formation of species from one common species that's at least initiated by a geographical separation. And you can see when they made a graph showing the number of islands, how many islands appeared how many millions of years ago. That's the green line on this graph. And they noticed that it corresponds quite well to a similar graph of the number of new species of finches over a similar period of time. And so it seems like that virtually every time a couple new islands appeared, that within a certain lag time, you can kind of see a lag time between them, that those new islands were populated by finches, which then evolved separately on that new island. So it's an interesting correlation. Now another type of speciation is called sympatric speciation. Sympatric speciation is different than allopatric speciation because you don't have to have any kind of geographic separation in order for it to occur. A good example of sympatric speciation is polyploidy in plants, where you can have two plants that are similar but not the same species. They can cross-fertilize to make a hybrid, and then the hybrid can go through a chromosomal doubling event to produce a fertile hybrid, which is technically considered a new species. That can occur exactly in the same place as the original parental species still live. Another example in the animal kingdom is the maggot fly, where maggot flies typically live on hawthorn trees, but when apple trees were planted in some of the same areas as hawthorn trees, a group of maggot flies apparently migrated over to the apple trees, and then the two groups stayed separate long enough that the flies on the apple trees became a different type of fly or different species. So now you have a different species of maggot fly that lives on hawthorn trees, and nearby you have a related but different species that lives on the apple trees. So this figure shows a phylogenetic tree, or an evolutionary tree. And evolutionary trees show relationships between organisms. It's based on a lot of data. This one happens to be based on a comparison of genetics or gene data. It's also sometimes built using fossil data or other kinds of data. And on this evolutionary tree you see Equus, which is the horse family. It includes horse, zebras, and donkeys. What you notice here on the x-axis, what's implied is always time. So 0 on the far right here, 0 means now, and as you go from right to left on the x-axis, there's an implication that you're going back in time. So for example, these two types of horses, if you take these lines here, these branches, and you trace them back to where they meet, then that point is called a node, and that node represents the common ancestor of these two horses. And so if you go back in time a certain amount, you would if you had a time machine, you'd be able to find the common species, the species that is the ancestor of both types of horses. On this phylogenetic tree, they've overlaid this sequence divergence, meaning these two types of horses have only about 1% or maybe a 0.5% difference in their DNA sequences. Now, usually they're not sequencing the whole genome, they're sequencing certain genes and using those as a kind of an estimate. If you go back further, though, on this line, you get to this node, which is, I don't know, maybe 7% sequence divergence. And so 7% sequence divergence would represent the amount of DNA difference between, for example, the domestic horse and the mountain zebra. Because if you go backwards on the mountain zebra line and trace it all the way back, the place where you first meet up with the line from the horse is right here at the 7% sequence mark. So that would indicate that those two organisms have this, their DNA is 93% the same, or at least the genes that they sequenced, it's 93% the same and 7% difference. The idea here is that the things that are only related by a distant ancestor are going to have more genetic differences when compared to each other. Things that have a more recent ancestor are going to have less differences when compared to each other. So, and that's a pretty reasonable assumption. Now, what kind of genes do they look at when they're making these comparisons? This chart shows, for example, in mammals, they would pick certain genes that are common for mammals. So actin, albumin, the globin genes, histone, things that are really fundamental, insulin, genes that are really important to the function of both types of animals or both groups that are being compared. And then they literally get samples from different organisms and line them up and see how many actual DNA differences there are between them. And they classify the differences that they find into two categories, synonymous and non-synonymous. So these are substitution mutations. Synonymous substitutions mean the same thing as the term silent mutation. So a silent mutation is one that doesn't change an amino acid in the resulting protein. A non-synonymous mutation would be like a missense. It would change an amino acid in the resulting protein. So you can almost change these headings to read missense mutations and then silent mutations. So missense is non-synonymous, silent is synonymous. So the missense mutations you see occurring at a lower level than the silent mutations. So in other words, for alpha-actin, you only see in modern organisms an occurrence of 0.01 non-synonymous mutations per nucleotide per billion years. That doesn't mean that non-synonymous or that missense mutations don't happen as often. What it means is that when they do happen, the organism that gets that mutation is less favored for survival, and so that guy dies out. And so in live modern organisms, you don't find very many that have any kind of mutation in those genes, because we say the gene doesn't tolerate the mutation. What it means is any gene that gets that mutation, that's going to give a disadvantage to that organism. So anything that's alive today is a survivor. So anything that survives is typically not going to have a mutation in these really important genes. So that's why these non-synonymous numbers are really low. Synonymous mutations, these are silent mutations, so these don't affect the ability of the organism to survive because they don't change the protein at all. So that's why you see these at a much higher level. They occur at the same rate as the non-synonymous mutations, and yet they don't affect the ability of the individual to survive. So we say that the synonymous mutations are more tolerated by the species. It turns out you can actually map the mutations to different parts of each gene, and you can look at what part of a gene tends to have more mutations. Exons tend to be kind of the critical part of the gene, and the lowest mark you have here are the non-synonymous mutations in exons. So the missense mutations in exons are the least tolerated. They are not passed on in evolution. Typically that's because the originator of the mutation dies or does not reproduce. So we say that important sequences are the ones that stay the same over time in evolution. If you look at something in human and something in dog and you see that the genes are identical, that probably means that gene is really important, and any mutation would probably give a really big disadvantage. So only the non-mutant alleles or the alleles with silent mutations are able to go on and survive. So what we try to do with all this data is put together a relationship between the mutation rates and the evolutionary divergence, and we create this graph, and it's a nice linear relationship. It's always understood that this is an estimate of evolutionary events. So let's say you have certain organisms on your molecular clock. You have some data about shark and newt and echidna and dog. And you map that and it makes a nice linear relationship here. So you draw the line and then you find a new organism, all right, I don't know, chicken. And what you're able to do is then compare the chicken DNA to some of the DNAs that you have on your chart, and based on how many differences you find there, you can then figure out how far apart it, how far back it diverged. And so you basically interpolate along this relationship. When you find new organisms, you can interpolate where do they belong in the evolutionary tree based on how many differences do they have compared to the animals that you already have studied. So it's a way of adding things into the evolutionary tree based on what's already there. It's always assumed to be an estimate of how these evolutionary events happened. Again, a hypothesis based on the data we have right now. But the molecular clock is based on, again, the same idea that the more closely related two organisms are, the more recently they shared a common ancestor. The more distantly related two organisms are, the more distantly they shared a common ancestor, and therefore distantly related organisms will have more genetic differences between them than closely related organisms.